Good morning, everyone, and welcome to the 2020 Layman Conference. We're very happy to have you here. We're excited for the conference this year. And obviously, we're doing things a little bit differently this year. So we are hoping that you will enjoy the presentations that we've put together. This is the pre-conference workshop for the Veterinary Diagnostic Lab. This year, we thought it might be useful to take a step back and provide an opportunity for practitioners to get more grounded on methods that we use from the molecular field for diagnostics. And so we have a broad range of speakers that will be walking us through some different methods that we use to help you understand why we use them, how you can use them effectively, and how to interpret and manage the results. And so I won't spend more time on the background. I do want just to provide a little housekeeping. If you have questions along the way, you can ask them in the chat box. And so we need to have you type them in, and then we'll try to get to those at the end of presentations. So we'll be going along and allowing the presenters to get through their presentations and then field questions at the end. Hopefully we'll have time after each presentation for that. And then we'll be taking a break at 10 o'clock and coming back at 10:15 for the second round of presentations. But first, we want to start with an overview of next generation sequencing, NGS as it's known. And so we are very fortunate to have Dr. Sunil Moore here with us this morning. He is the head of our Molecular Development Laboratory section. He got his DVM equivalent, so it's the British version, BVSC, in Hisar, India in 2006, a master's there as well in veterinary epidemiology and preventative medicine. And then he did his PhD work here at the University of Minnesota, and since 2017 has been here at the Diagnostic Lab as the head of our Molecular Development section. So Sunil, we're excited to have you walk us through some background so we have a better understanding of what next generation sequencing is all about. So Sunil.
Thank you, Dr. Torreson. Good morning, everyone. So as we all know that sequencing has touched all sectors of the research field, even in the space also. But for us, the question is why I should request for NGS, or why we consider NGS as a diagnostic tool. So to start with that important question is, the first and most important, if there are clinical signs or this, but we do not have any information, or the clinical samples are negative with all our PCR test or the routine test, so then the only option is to consider next-gen sequencing to try what's present there. Second, if EM or histopath, they indicates some viral etiology and it's negative by PCR test or virus isolation. Sometimes we see the uncharacterized CP in the cell culture. So virus is growing, but it's negative with our usual test. So then what this virus is growing in the cell culture, NGS can help to characterize this unusual virus. And then obviously detection of novel divergent emerging pathogens such as the SARS-CoV-2 is the recent example that helps with the NGS to characterize fully. And in the disease syndrome like respiratory syndromes or the enteric syndromes cases where we have the multiple factors involved in the disease, then NGS helps to look at the totality of the pathogens, virus, bacteria present instead of doing the individual or the single PCR for 5, 10, 15 pathogens. And whole-genome sequencing to know if the virus or the bacteria is related to this vaccine strain or the different from the strain, or is related to the previous outbreak or the different. So all these cases, next-gen sequencing is very helpful. So here is the basic overview of the NGS. Like any other molecular methods, here also it starts with the sample processing and nucleic acid extraction. But then other important step here is to prep the sample for sequencing, which is called library preparation, and then sequencing on the different sequencing tools, equipment. And then another important point is the data analysis. So NGS is divided into two main components, the wet lab, sample prep to sequencing, and then bioinformatics analysis. So here is the overview of the sequence analysis. It's very complicated and long process, but ultimately we just see the final output, like how our sequence is relates, or it's a new virus or new pathogens, and to make the decision based on those results. So just to start with the basic terms, we have these metagenomics or microbiomes are interchangeable, being used very commonly. But overall, the microbiome is looking at the total community present in the sample, which may be the bacteria, fungi, viruses, and then accordingly it's called the Bacteriome, Virome for viruses. So to start, it's a broad overview, but just to simplify how we use the NGS or characterize the NGS at the diagnostic labs or in the veterinary diagnostic, there are two main methods. One is called targeted NGS, or where we enrich the sample for particular virus or the bacteria. Two example is like 16S NGS and influenza also, where we have the specific primers to amplify our regions of our interest. It's like a PCR, but then using the NGS methods, it's amplified to generate the millions or billions of reads and then do the data analysis. Second method is non-enrichment, or the non-targeted or unbiased amplification of whatever present in the sample. So that's what it comes to the virus or pathogens discovery or the disease investigation, where we do not have any prior information about the pathogen present in the sample. And whole genome sequencing also, if like influenza, if we do not have the specific primer to amplify the whole genome. So in that case, no specific primers to amplify. So it's just attached with all the nucleic acid present in the sample. So like in this case, the Illumina is one example here, and then it will amplify everything present in the sample. So just to touch two most common things here, like to study do the NGS on the bacterial or for the bacterial identification. It's called Bacteriome analysis. So the most and the common or the easy to use is if we have the pure culture. So we know that what type of the colony or bacterial colony we are going to amplify doing the whole genome sequencing. So in that case, just pick up the bacterial colony or the culture, extract the DNA, and then do the NGS library prep and the sequencing, and then do the analysis to assemble the whole genome to compare with the previously reported sequences or to find out the information that we are looking for. Then the two methods on the clinical samples. So obviously there will be a lot of materials in addition to our bacteria of our interest. Then in that case, one method is the targeted, as I mentioned. So 16S, just amplify the region of our interest and do the library prep and sequencing. So it will just give the bacterial diversity present in the sample. Second is the non-targeted method. And as we know, in the sample, there may be present so many different things, including our bacteria of our interest. So in that case, it will amplify everything. And then with the bioinformatic analysis, we figure out how much the sequence is related to our bacteria of our interest and what are the other junk or the non-related things present here. Same in Virome analysis also. As we know, the sample have everything, including the viruses, and viruses seems to be or known to be related in the less abundance or concentration as compared to the host genome or the bacteria. So we have to do the enrichment for the viruses to get the better results for viruses, which is called different steps, different protocols have been optimized, but commonly they are known as centrifugation, filtration, nuclease treatment. So a lot of preparation steps before going for the RNA or DNA extraction. So it's important to know what our objective is for doing the NGS, so that these different steps can be applied accordingly. So just overview of the library prep steps. Different parts are involved here, but nowadays the commercial kits are available to simplify and to shorten the time for the library prep. So it's going shorter and shorter. So that's what also the time consuming part for NGS. And then once the library is ready, then it's loaded on these flow cells on the different types of the sequencers. And these are the just rows where all these millions or billions of sequences are generated. It generates very large amount of the data depending on our sequencing run or then sequencer. So here is these flow cells, these dots where the sequencing occurs, and we got this sequence data in the FASTQ zip files, which the bioinformatics starts from this data. So different steps are involved in the bioinformatics. Again, it's a challenge to find out the most important information out of these millions or billions of reads. And here is which is considered as the most challenging part nowadays. With the technology, it's easy to sequence, but it's challenge or the complicated to find out the relevant information from these millions or billions of data. So we have invested in this bioinformatics side also, both on hardware and softwares, to do the rapid, fast analysis, as well as to secure, store your data securely. And these are the different commercial tools or the pipelines, which we call, where we have all the things are assembled mainly in three parts, like pre-analysis to clean the sequence, and then do the assembly, and then final output where we see the final results in the phylogenetic tree or the AMR pattern, or how it relates with the vaccine or other sequences. Here is our VDL pipeline, where we have very robust analysis to make sure that we provide the quality data. And here is the final endpoints which we share with the pathologists or the veterinarians. So the common term in NGS is garbage in, garbage out. So if we do not have the good quality sample or then good quality and concentration of the nucleic acid, then even after having the millions, billions reads, we may not be able to get the good quality information out of it. So our diagnostic lab, we have optimized protocol to work with all different types of the samples with different species. And we have the MySeq, MinION in-house, and then we use the NextSeq or the NovaSeq from our genomic center. And once we have the data, then we do the identification of the sequences to find out if it's a new novel pathogens or the vaccine-like virus or bacteria, or what other information that pathologist or veterinarian wanted to know out of doing this sequence on the clinical samples. So again, it's divided into two main parts. One is the NGS to use as a diagnostic test. As I mentioned previously, if we do not have any prior information about the pathogens, all PCR tests are negative, or some indication from EM that it's maybe small round virus particles, but it may be any Astropicorna or Calici type, and some viral lesions indicates that the virus is present there. So in that case, NGS helps to figure out what virus is causing the disease or associated with the disease or the outbreak. Second is to characterize the pathogens. So in that case, like the PERS, if we have the PERS PCR positive, but we wanted to know, or the by Sanger, it relates with the vaccine based on OR5. So we wanted to know whether it's a different sequence or the related to the vaccine, or is there any insertion deletion. Then Whole-Genome Sequencing helps here to differentiate from the previously reported sequences. And third is that sometime if we also discuss the specific problems and or the research topic, or like the survey of a farm to do the screen for the bacterial pathogens or the microbial community. In that case, also we happy to discuss the specific project and plan the sampling and the testing accordingly. So with that, our diagnostic lab offers the different types of the NGS services for starting from bacterial whole genome sequencing from culture to the metagenomics to amplify everything present in the sample, or to focus on the DNA virome or the RNA virome analysis. And then we also offer the service if there is a request in the bulk, like 15 or more isolates for the sequencing, provide the reasonable cost for the different types of the NGS request. So with that, I would like to thank my development team and all the veterinary diagnostic lab sections and the pathologist to support for these NGS services. So with that, I think we have the few minutes to answer any questions.
Thanks, Sunil, for that presentation. That's a great start for us to get an understanding of the tools and the complexity of the work that's done. Nobody has listed any questions on the chat, and so this again is an invitation to people, if you do have any questions, to type them in the chat space next to the presentation. I would like to ask, so we've become accustomed to people, producers and veterinarians submitting samples, have pretty high expectations for what we can do with samples for PCR testing. Are the sequencing methods subject to any additional challenges? In other words, what can people do to submit the best possible samples if they want sequencing performed?
Yeah, so there is another presentation on this NGS successes and challenges. So I will discuss in detail there also. But as I mentioned here, this NGS, the common term is garbage in, garbage out. So if we do not have the good quality sample, so that means is like if the sample is collected but not stored properly before requesting for NGS, then we may not be able to find or our results may be biased. So like there's some enzymes like RNase, which degrades the RNA very easily. So in that case, our results may be biased. So it's the proper collection of the sample and the proper storage before requesting NGS is very important. And I would suggest like I'm available and our pathologist also available better to discuss and plan the project or any sequencing work because it's very expensive. So it's good to have the good pre-planning before going for the NGS request so that we can increase our chances to get the good quality data.
So now the floodgates have opened, Sunil. We've got a lot of questions, and so we'll spend a couple more minutes on them. One from Ed Ackerman is I think pretty straightforward. What is the difference between NGS and WGS? Can you clarify that?
Yeah. So NGS is like next-gen sequencing is the common term that we use for any NGS platform. So next to the Sanger sequencing, we just common use NGS. WGS is the whole genome sequencing. So if we are doing like just requesting for the whole genome of the purse, whole genome of strep suis, then it's the common term is like WGS. But in NGS, suppose if there is a disease investigation case, we just wanted to know what virus or bacteria is present. So in that case, we will say, okay, I wanted to go for NGS to find out what virus or bacteria is present in my sample causing this particular disease or the clinical signs or the lesions. In that case, or the EM is showing some small round virus particles, let's say, then we will say, okay, let's do the NGS to figure out what virus is or the virus is present.
A couple more follow-up questions on sample submission. Are there specific media that are better for sending in samples? And In a related question, so for example, for bacteria, why would we use NGS if we already know a sample is PCR positive, for example?
For sample submission, I would say like for our other test also, if we can get sample like shipped overnight or the in relatively short amount of time, then we will prefer to process samples for NGS in our lab. So any standard PBS will be fine. Sometime in the in other media, I will show in another example in the next presentation, but in some media like FBS or donor host serum related medias, there may be some adventitious or contagious virus present, so which may affect our NGS interpretations. So second is about the bacteria. So in our one more session on the bacterial genotyping, we will explain in details also, but sometimes it's important to know like how this particular bacteria is or the colony is really true to that. I'm sure now test can go for the good confidence to say this is the really that particular bacterial colony. But if they are very related, like erysipelathics, rugiopathy, or the tonsillorum, then how to differentiate these two? Or there was one example to differentiate the one vaccine strain versus the field strain, which we will explain in the one of the presentations. So there we cannot tell with the culture, but with doing the whole genome sequencing, we can clearly differentiate the two strains.
Well, thanks. You remind me that we are. It reminds me a little bit of Saturday morning TV, where people would say, "Stay tuned for the next episode." So that's a good lead into some of the presentations we have coming later on. So thanks, Sunil. We appreciate your presentation and look forward to more from you later this morning. So thank you very much. In order to stay on time, I think we'll try to address some of the additional questions in the chat room through the chat room, or circle back and address them later on. But now we'll keep going with our presentations and look at some specific applications of NGS that are for one of our still most important viruses, PRRS virus. Dr. Albert Rovira is the assistant director here at the Diagnostic Lab. He's been working on PRRS for a long time. He did his DVM and master's training in Barcelona, where he hails from originally, and did his PhD work here at the University of Minnesota, and has been working here at the Diagnostic Lab for a number of years, and continues to perform research in a variety of areas, including some of these molecular methods related to PRRS virus. So Albert, thanks for helping us out this morning, and go ahead and help us understand how we can use these tools in clinical situations.
Thank you, Jerry. I'm going to share my presentation here. Welcome everyone to the Lemon Conference. It's a different Lemon Conference, but welcome everyone. It's nice to see that the questions are coming in the chat, because otherwise I didn't know if you were there. I'm sure a lot of people are there, some of you in your pajamas. But we're going to talk about one of our favorite topics here, PRRS, and we're going to talk about PRRS whole genome sequencing. So a little bit about what do we do once Sunil is done with all his lab work and with all his bioinformatics. He comes with a PRRS whole genome sequencing. What can we do with that? And this applies to other viruses, but of course, PRRS is the one that we more commonly deal with. So why sequence the whole genome? Sunil talked a little bit about this, but specifically for PRRS virus, why sequence the whole genome? That would be the first question. And the most basic answer is to answer the same old question of is this the same strain? So we're usually trying to compare one strain to something else, to our neighbors, to the strain in the farm, to a vaccine, to something else. So basically, this happens when the answer from the OR5 sequencing is unclear. So those cases in which the answer to is this the same strain is a maybe. When you don't get 100% similarity, you don't get completely different, something in between. or when the answer from OR5 sequencing doesn't really match the clinical presentation. So you get a sequence that it matches something that you knew it was pretty mild in the past, and now it's acting very virulent, or situations like that. And now keep in mind that OR5 represents only 4% of the PERS genome. So in those cases where OR5 is really not giving us an answer that is helping us, it makes sense to go dig deep in the genome of PERS virus, because there's a lot of it that we're not seeing. So very quickly, let's review the PERS genome structure. It's a single-stranded RNA virus. I call it here medium size, and on the right-hand side, you can see a comparison with PCV2 and ASF for viruses that we might be familiar with. And so you can see there the length of PERS in that vertical line. PCV2 is about 10 times smaller, and African swine fever virus is about 10 times larger than PERS. So that's why I said that it's about medium size. And that's important because when we get a whole genome, it's manageable. We will be able to interpret it. It's complex, but it will be manageable to interpret. So if this black horizontal line represents the whole genome of PERS virus, that little box right there at the end, that represents the OR5 that we are also used to sequence and to interpret. And there's a lot more of the virus, as you can see here, and many other genes that we are just not seeing. Excuse me. This is just to stress again that we're only seeing a little part when we do OR5 sequencing. And the other characteristic very important for interpretation of PERS is that it's high genetic variability. Most of you will have one of these charts for your farm, for your system, for the farms that you visit with PERS sequences. And you know that even within the North American PERS virus, there can be a lot of variability. Some of these strains are 20% different in their genomes. So there can be a lot of variability, and PERS changes a lot. And that means also that the same strain is going to be changing, and there's going to be some background change that happens in the same exact strain as time goes by. So that's going to be important for interpretation. Now, a little bit of comparison of OR5 versus whole genome as it is today. First of all, we already talked about this. OR5, we're only sequencing 4% of the genome. Whole genome sequencing, obviously, we're covering 100% of it. I think by now, you probably remember that 4% number. I said it many times, but I think it's important to realize how little we're seeing of the virus. Turnaround time, OR5 sequencing, we're now offering 2-day turnaround time. That is very good. Whole genome sequencing, we're doing 2 weeks. Sometimes it gets more complicated and it gets delayed, but we're doing about 2 weeks. So there definitely OR5 wins. For cost, $100 for OR5 sequencing, between $250 and $350 for whole genome. So there, OR5 sequencing will win as well. Success, what I call here success rate, would be, we're pretty confident now. It depends a little bit on the CT value, but we're pretty confident when we do OR5 sequencing that we're going to get a sequence. And with whole genome, it's getting better, but there are still instances for different reasons in which we cannot get a whole genome. So that's what I call the success rate here. It's very high in OR5. It's what I would call medium and improving for whole genome. So we're working on that. And finally, for databases, again, here OR5 wins in that when you get a sequence, you want to compare it to what else is available. And we have a lot of OR5 sequences available, either in the diagnostic laboratories or in public databases. So we have excellent databases to compare with. For whole genome, we haven't been doing it that long. And so obviously the database is to compare with, it's not as good. But the one thing that I want to stress here is for OR5 sequencing, we're pretty much maximize that technique. We cannot make it much better. And for whole genome sequencing, all these areas in which I said OR5 beats whole genome, we're working on them, and we're very confident that we're going to get those better. So it's going to get faster, it's going to be cheaper, it's going to get more reliable. So this is as it stands now, but definitely there's a lot of improvement to come with whole genome. So what I'm going to try to do is show you 3 cases in which we did whole genome sequencing for pairs, and hopefully that will show a more practical application of what this technique can do for you. The first case, some of you might have seen it. I presented it in Lemon last year, but it was a small group, and I thought it would be good to review it because it has some good take-home messages. This case was a case of abortions in a PERS-stable sow farm, and 10 serum samples were collected from aborted sows, and they were all positive for PERS. CTs range from 23 to 39. So we went ahead and sequenced the OR5 of one of those, and actually we ended up sequencing the OR5 of 7 of these, the ones that have a low CT. And what we found in all of them was the same sequence, the same OR5 sequence, and it was 98% similar to PERS vaccine A, to a vaccine, one of the commercial modified live vaccines in the US. And so that was strange. We were not expecting to see vaccine in sows that just aborted, but it's even more strange because vaccine A had not been used in that farm for 2 years. So the question here was, is this virus really closely related to vaccine A, or is it a different virus? And the harder to answer question, but also in our minds, was, is it a lateral introduction? So what we did is we did grow a viral isolate from one of these samples, and we did whole genome sequencing in one of these samples. I'll show you now what we found. Now, when you do whole genome sequencing, you don't have to look at OR5. You can look at the whole genome, or you can look at the different, you can look at whatever you want, really, but you can look at the different genes one by one. This is what we did here. And I want you to take a look, to go backwards, take a look at ORF7. We see that it's very, very similar to vaccine A, 99%. ORF6 is 99.1% similar to vaccine A, ORF5A 99.3, ORF5 98.0, matches what we saw with the initial ORF5 sequencing, ORF4 99.6. So this is very consistent to what we saw before with OR5 sequencing. But then as you get closer to the first part of the virus, you see ORF3 is more similar to vaccine B, to a different vaccine, 98.0. And this is a vaccine that is actually used in this farm. ORF2B is 100% similar to vaccine B. ORF2, 99.4 to vaccine B, and ORF1B, 98.5 to vaccine B. ORF1 didn't match very well either. So, ORF1A, I meant. So what I'm seeing here is the first part of the virus is more similar to vaccine B. The second part of the virus is more similar to vaccine A. So I started thinking, well, maybe this is a recombinant that has one chunk from vaccine B, one chunk from vaccine A. So we did a recombination analysis comparing our genome with the genomes of vaccine A and vaccine B. And this is what we found. I want you to focus on this line here on top that has parts of them that are green color and parts of them that are purple. And remember, I thought that maybe this was a recombinant with one part one vaccine, another part the other vaccine. This actually has 4 parts, 4 different segments that are that come from vaccine B, and 4 parts that come from vaccine A. So it's a lot more complicated than I thought, but certainly it did match recombination. It did suggest that this was a recombinant virus between these 2 vaccines here. So this is a virus that probably has been circulating long enough in the farm so that it has been capturing, or both viruses have been circulating long enough that a recombinant with several segments in it has been created. So what's the interpretation of this case? First of all, remember our question was, we wanted to know, is this vaccine A? And the answer is no, this is not vaccine A. Now that we get to see the whole genome, we can tell that this is not vaccine A. Then what we are learning here is that multiple strains of PRRS coexist in swine populations, and they may result in new strains through recombination. This is not something new. We've known that PRRS can recombine, and new strains can be created through recombination. We're just learning more and more about this and trying to figure out how common this is. And finally, with this case, we see that recombination between vaccine A and vaccine B may have resulted in a virulent strain. And note here that I'm saying it may have resulted. Certainly, the clinical signs suggested that the finding this virus in every single animal that we tested that had aborted suggested that. There's a few things that I would like to see to be even more confident that this virus is really a virulent strain. Of course, I would like to put it into pigs experimentally and make sure that it causes disease. And I always like it when I get the virus in cases of abortion actually from the aborted fetuses. It kind of gets you a little bit closer, one step closer to causality. In this case, it came from the serum of the sows, but still, in a herd that is under control or well vaccinated, you wouldn't expect to see all these positives in sows anyway. So that's what is suggested for this particular case. So that was a first case, a case in which whole genome sequencing clearly gave us an answer to the question that we have. And that's why I wanted to show it. And it also had the recombination component to it. So a very clear case. Not all the cases are going to be like this. Not all the cases are going to be so straightforward or so revealing. So we're going to see a couple more cases that are going to highlight other situations. Case 2 is it starts pretty similar. It's an abortion in a PRRS stable sow farm. In this case, there's a 3% increase in stillborns. There is a 5% increase in pre-win mortality. And in this case, there's 7 serum samples that get sent to the lab from aborted sows. In this case, they get tested in pools, in 3 pools, and 2 of the pools are positive with CTs of 24 and 33. 1 pool is negative. We do the OR5 sequence, and we get 99% similarity to PRRS vaccine B in this case. And this vaccine, it is used in this farm. It is used in gilts. But again, we didn't expect to see a vaccine sequence in a case of abortions in sows that just aborted. So again, the question here is this virus really closely related to vaccine B? Or is this a different virus? Is this one of these recombinants? If we look at the rest of the genome, are we going to see that this is really something else? It's really a field virus. And finally, is this a lateral introduction? Again, that's a harder question to answer, but that's always in our minds. In this case, virus isolation was negative, so we did whole genome sequencing from one of the serum samples that was positive. So for this case, the results that we found for OR5 sequence, remember for the OR5, it was 99% similar to vaccine B, right? For whole genome sequence, once we got the whole genome, when we compare it to vaccine B, it was 99.3% similar to vaccine B. So the results that we're getting from whole genome are very similar to the results that we were getting from OR5. And this is how, like I told you, this case is different from case 1. In this case, we are not finding something completely different. The sequencing of the whole genome, it's kind of confirming what we are seeing with OR5. We did a recombination analysis. There was no evidence of recombination. And it's always good to look at those differences. So since it's 99.3 similar to vaccine B, it means that there is 0.7% of the genome that is different between these 2 strains of PRRS. And there's many ways to represent this many different types of software. But for example, here you have this representation. If you look at this gray bar, this gray horizontal bar that represents the genome of PRRS virus. On top, you have a scale that tells you the nucleotides. So those are the 15,000 nucleotides of the genome of PRRS virus. So you know where you're at. And then all these little ticks, all these little red vertical bars or lines are nucleotides where there is a difference between our vaccine, between our new sequence that we just generated and the vaccine. And so what this is telling me is that these differences, these nucleotide changes are all over the place from the beginning of the virus, middle, end of the virus. Some areas are naturally more likely to change. There are some areas in the genes, in the proteins of the virus are more likely to change, but still, it's not like all the 0.7% of the changes are all concentrated in one area. So I think it's, even when you have very little change, it's important to know where your changes are in case they could be telling you something else. In this case, they look randomly distributed. So the interpretation, the strain is 99.3% similar to vaccine B. That's an interpretation, that's a result, but the interpretation here would be a vaccine-like strain appears to be associated with abortions in this farm. Now, all the caveats that I mentioned before, for the case before, still apply to this case, even more so, perhaps, since we had 2 out of 3 pools that were positive, 1 pool was negative. Again, these samples came from the serum of the sow, not from the fetuses. So that's why I'm saying that it appears to be associated with abortions. Still, we would want to get more proof to that. But if that was the case, if this strain really is able to cause abortions, a next step could be to figure out why is this strain that is so similar to vaccine B able to cause abortions. There are 105 mutations, that's the 0.7% that is different. And are there any mutations that have a significance that are making the strain different, if that is the case? And that's something that I wish in the future, with this whole genome sequencing work, we can answer this type of questions. This gets into the virulence of the virus. Understanding the virulence of the virus, it's something that we don't know very much now, but as we generate more information, I'm hoping, and with more and more research put into it, I'm hoping that in the future we can answer that question. I don't think that we're there yet, but it's something that we would like to work on. Now, I've been talking about vaccines a lot, and so this is not a talk about vaccines, but there's a lot of these cases that involve somehow vaccines. So I wanted to talk a little bit more about vaccines, give some considerations about vaccines and why we tend to talk about vaccines so much when we do whole genome sequencing. First of all, they are more likely to get sequenced, to get put into whole genome sequencing, in part because if you get a clinical case and you get an R5 that suggests vaccine, that is unexpected. So that's one of the things that triggers whole genome sequencing. Sometimes also because there's financial support from the vaccine companies supporting these investigations. But also they are more likely to get compared. So they are more likely to become one of the strains that you use for comparisons when you obtain a whole genome sequencing, whole genome sequence that doesn't look like vaccine, but you might want to compare it to it. Because why? Because it's one of the known sequences in the farms. A lot of the farms are vaccinating, so we want to include that if the farm is vaccinating, and also because the sequences are available. There are some cases where we would want to compare to other sequences, but those sequences are not available. Some of these vaccine sequences are available, so that's they get compared to whole genomes that we generate. But also a common question is, are they more likely to create recombinants? I don't think they are more likely, or I don't have evidence to show that they are more likely to create recombinants per se, but maybe they are in the sense that they are very, very abundant. They are very, very common. Vaccination, at least here in the US with modified life, is very, very common. If you think about it, most of the pigs get a vaccine at some point in their lives, and so it's more likely that at some point, if there is a recombinant, it might involve one of these viruses. That might not be a bad thing, getting a recombinant with a vaccine strain as opposed to getting a recombinant with two wild strains. But anyway, that I think that it's part of it. The other part of it is the way that we use vaccines. It might increase the likelihood that they end up forming recombinants, especially when they are used in, for example, mass vaccinations. There might be some pigs that are viremic when we inject them with vaccine. We're also injecting that vaccine, so we're bypassing some of the natural immunity of these pigs. And remember, to create a recombinant, you need two different strains of a virus in the same cell at the same time. So that's a very unlikely event, but by doing, for example, mass vaccinations, we are making that a little bit more likely that it will happen. Again, I'm not saying it's a good thing or a bad thing. I'm saying that it might be more likely because of the way that we're using it, if you were using it that way. And finally, rotation of vaccines. In some farms where we've seen these recombinants with vaccines, we have observed that they were rotating vaccines, and so two vaccines that had been rotated recombined. Rotation of vaccines, I think it makes sense if you want to create broad immunity. Now, it makes more sense perhaps with kill vaccines than with modified live vaccines, because you also introduce a little bit of the risk that you would create a recombinant if they happen to be circulating at the same time. And finally, let me state this because I get questions about this. I have no reason to believe that any vaccine is more likely to create recombinants than the rest. So we have several several modified live vaccines available in the US, and I'm not naming them here. I'm calling them vaccine A, vaccine B, vaccine C, exactly because I don't think one is more likely than the other. They are live, and this can happen. And I've seen cases with at least 3 different vaccines from different manufacturers. So the point is not to talk about which vaccine. The point is to talk about PRRS viruses, and live vaccines are PRRS viruses. So I think it just applies to them as well. And finally, you're going to see probably more of these as we use more and more next generation sequencing, and we apply all those techniques to whole genome sequencing. We're going to see more cases of these recombinants with vaccines. These are 3 examples of recent papers that came out on modified live vaccine, either recombining with another modified live vaccine or recombining with a field virus. One is from Europe, one is from Asia, one is from North America. So you get the idea. We're going to be seeing these publications, this information. I think it's a good thing. I don't think it necessarily means that it's happening more often. It probably means it reflects that more and more labs are able to do whole genome sequencing and to uncover these events that were probably happening in the past anyway. All right, so we're going to move to the last case. In this case, it's not going to involve vaccine. So hopefully here you'll have an example of how this doesn't necessarily involve vaccine. And again, this is not going to be a perfect case with perfect answers like the first case that we saw, because that's a reality sometimes. And this was a PRRS outbreak in a sow farm. This one was a 174 virus. The farm did serum inoculation, herd closure, and then they went negative for 8 months on processing fluids. They also did individual piglet samples, more than 180, and they were all negative. So they were good to go, ready to open the farm, and then they had another PRRS outbreak. And it wasn't a mild one. 3 weeks of abortions, double sow mortality, increased pre-weaning mortality. They sequenced the virus, of course, and this virus was 98.5% similar to the previous virus, and it was also 174. So is this the same virus? The first outbreak and the second outbreak, is this caused by the same virus? If it was the same virus, how come the second outbreak is so aggressive? If all the animals were inoculated with that virus, they should have homologous protection. Is this a lateral introduction that just seems very similar in OR5? The 174 viruses are pretty common, so it could be. So that was those were the questions that we were trying to answer in this case. So with the whole genome sequencing of one sample from the first outbreak and one sample from the second outbreak. For the first PRRS virus outbreak, we only got a partial genome sequence. We couldn't get the whole genome. Actually, what we got is something like this, little fragments here and there. And those were 9 contigs or fragments that we could put together. And they range from 118 to 370 nucleotides. And that accounts for about 11% of the genome. For the second PRRS break, we got the whole genome sequence. So we were good there, but we were a little disappointed, right? We were saying that for OR5, you only had 4%. Well, now you have 11%. You're not that much better off. A little disappointed, but we should not stop there. We should try to get as much as we can out of this information. So remember, OR5 comparison for these two viruses was 98.5 similar. Now, when we compare these 11% of the genome that we have to compare now, we have 94.2% similar. So that's starting to tell a different story. But now it's very important to know where these fragments are. And so I'm going to show you here. At the bottom, you have the whole genome of the second outbreak PRRS virus in purple. And the top, you have the fragments and where do they fit, where do they match the genome, the fragments that were obtained for the first outbreak. And so we have 9 fragments. And then on top at the right-hand side, here, I think you can see my cursor, you have the OR5 sequence that we had from before. That is also from the first outbreak. And so the good thing here is we have some fragments that are at the first part of the virus, some are in the middle, some are towards the end of the virus. Now, when we look individually at each of these fragments, yes, they are small, but we can look individually at what's the percentage similarity of these viruses. We see that the 6 fragments that are towards the end of the virus, they are all very similar to between the first outbreak and the second outbreak. So that's 100%, 98, 96, 99, 98, 98, 99. So very similar. Now, when we look at the 3 segments, 3 contigs that we found at the beginning of the virus, those are very different. Those are telling a different story, 90, 86, and 82. So in summary, this first part of the virus is about 86% similar to the second virus, and this part of the virus, most of the virus, is 98% similar to between the two, the first and the second outbreak. So the interpretation of this case is that the first and the second outbreaks were caused by different strains of PRRS virus. Yes, most of the virus was very similar, but there is a chunk at the beginning that it's pretty different. That is different, and it's probably coming from a different strain. The differences between these 2 strains may explain the lack of protection. And this is, again, something that we hope that in the future we can do is extrapolate more from whole genome sequences to what that means for protection, for immunity. We're not there yet, but that's something that we hope that in the future we can do for interpretation. So this, at this point, is just speculation. Now, where did the second strain come from? We don't know. It could be lateral introduction, or it could be recombination. It could be a strain that was in the herd that recombined with the strain from the first outbreak, and we got this recombined strain. Remember that we did a whole whole herd serum inoculation. So who knows, maybe that might have helped create this new virus. How come we didn't see any of that during the 8 months that we were sampling and testing for PERS? It went undetected, so it could be, or it could be that it's a lateral introduction. It could be. And here's a situation where if we had all the history of the previous viruses in this farm, and when I say the history, I mean whole genomes of previous viruses in this farm, we would be able to compare to those previous whole genomes and maybe help us figure out if it's a virus that was created internally or it came from a lateral introduction. But we don't have that information yet, and we would like to start building that information. So that takes me to the end of the presentation. Hopefully, I'm not going too long, but we're getting towards the end. We're going back to this idea of when to sequence or why sequence whole genome sequencing. Before we talk about sequence whole genome do whole genome sequencing when the answer of OR5 is unclear. We cover that when the OR5 sequencing doesn't match the clinical presentation. We cover that. This is basically the three cases that we saw basically talk about this. But I would add sequence whole genome, the whole genome of PERS with each new outbreak. So you have a new outbreak, get your sequence, get it on file. Even if it's not helping you right away, build a strong database for future comparisons for the reason that I just explained for the last case. And these may or may not be similar, but sequence whole genome, the whole genome of PERS before each new intervention. In some cases, it will be at the same time. When you have a new outbreak, you'll start a new intervention, but that's not always the case. If you start a new intervention, sequence your virus before, because then if you have another outbreak later on, you will want to know why, if it's the same virus or not, if your intervention was correct and you just happen to get another virus, or if there was a problem with your intervention. So I would add those recommendations for whole genome sequencing. And then you can start creating a nice, strong database for your farm or for your system and create charts like this. So this is one of the systems that we have more information about. They're creating a really nice, strong database that gives them a lot of background. These are all whole genome sequences, and you can do more or less the same things that you do with OR5, only you can do that with any part of the gene and create this type of charts. So here you can start seeing that we can identify several groups, and some are very, very different up here on top. Those are vaccine-like sequences. We can also look at this. If you were not confused up to this point, maybe this will confuse you even more. But this type of data, you can look at them many, many different ways. And this is the same data, but you can look at them in this type of heat map. Here on the top left corner, you see that big red area. Red means more similarity. This is a group of strains that are very similar, and it would match the strains in the lower portion of the dendrogram. And then as you go down and to the right, you will see some more spots that are red. Those are other clusters of strains that are very similar. And then to the bottom right, you'll see a couple of small groups of strains that are similar. Those are the vaccine-like sequences that are similar to each other, but they are not similar to anything else. So another way to represent this, we're creating a lot of information that hopefully this is going to help us with the new outbreaks, just to go beyond the OR5. So potential applications of whole genome sequencing, we basically cover strain ID / tracing. That is what we covered in case 1, case 2, case 3. But we hope, and we're working on trying to use whole genome sequencing in the future to understand and predict virulence, and also to understand and predict the immune response or cross protection. That would be great. We're not there yet, but definitely that's something that I hope that we can add to the value of whole genome sequencing. And finally, and related to that, I wanted to highlight we have a great group of researchers here at the College of Veterinary Medicine in Minnesota working on whole genome sequencing and trying to answer those questions. So we have molecular biologists working with immunologists, working with epidemiologists as a team, trying to answer things like genome diversity from the pig level, the quasi-species of virus that coexist in one pig at the same time, to the litter level, to the farm level, all the way to the region level, and the big changes in strains that circulate, over time and in different regions. So the whole spectrum of genome diversity. And then we have studies looking at burst transmission and immune response and cross protection that are going to help us with that type of interpretation. And we have studies looking at improvement of burst diagnostics that is going to make whole genome sequencing cheaper, better, and faster. So that's what I got. I wanted to thank the whole team of diagnosticians here at the Diagnostic Laboratory, and especially Sunil and Vikash, who work on all this behind the scenes and get all these sequences put together, and also the veterinarians and producers that that helped me with information for the 3 cases that you saw today.
Thanks, Albert. That was a great presentation. Obviously, not only an incredible diagnostician, but also a great teacher. So that was very helpful, I think, for people to understand. Partly, don't try this at home. This is a fairly complex thing, but also that it provides useful, accessible information that can be applied in the field. There's been a robust discussion in the chat room. We don't want to steal more time from Matt, so I think we'll let it go. But I will steal from Matt just for one question, because I think it's important in this context. What bias is there in terms of either selecting the low CT value PCR sample in order to have the best odds of having a successful sequence? Or Sabrina Swenson was asking, what if you do a virus isolation and have additional bias from what will grow in culture? So what factors cause that bias, and how can we get to the truth of what's going on most effectively?
Yeah, the ideal situation would be to sequence the sample, the original sample, not the viral isolate. That would be ideal, because then you wouldn't you would get the real sequence as it is in the pig, and you wouldn't capture any of the changes that happen in the virus once you start passaging it on cells. And whole genome sequencing is getting better and better, so we're getting more confident that we're going to be able to do that routinely in the future. I like to, at the same time, try to get a viral isolate, because there's been cases where I got a partial genome from the sample, but from the isolate, then I could get a full genome. Again, as the technique gets better, I think we'll be more and more confident from the original sample. The original sample might contain other viruses in it that we're not aware of. And then what happens is they basically, we start sequencing those viruses, and they take away some of the power of the next-gen sequencing, and we end up not having, in some cases, the ability to sequence all the pairs that we wanted, because these other viruses take over some of the reagents in the technique. But ideally, we will go with the original sample, but it's always good to have a backup virus isolate.
Okay. Thanks, Albert. I think we'll move on, and we'll continue the discussion in the chat room that has a pretty significant list of questions that we'll be wanting to address. And Matt Sturos has been the yeoman answering a lot of these questions, but we need to let him do his high wire act now without a net. Matt is a diagnostician here at the Veterinary Diagnostic Lab. He's also a graduate student working on Seneca virus with Fabio Vannucci, and also is a board-certified pathologist. He's originally a Uper, comes from the Upper Peninsula of Michigan, did his DVM training here at Minnesota, went out into mixed animal practice, came back for a pathology residency program, and now is working while also trying to conquer a PhD program. So he's going to help us understand what you can do at home. Albert showed you what maybe you shouldn't try at home. Matt's going to walk us through various molecular methods that we think can be useful for people to do. And so there are a number of links posted in the chat room for people if you want to play along at home or wherever you are. And Matt, we look forward to your presentation on some of these methods that can be done by themselves, right?
All right, well, thank you for that introduction, Jerry. Welcome everybody that's tuning in, whether it's live or if you're going to watch this after the show. I'd say good morning, but I'm not sure what time zone you're in, so I guess good day would be the appropriate greeting. So this is meant to be a hands-on workshop. I'm going to give some basic information. If you're already a molecular epidemiologist, you might not need to stay tuned for this part, but if you wanted to learn more or refresh your memory about sequences and sequence manipulation that you can do yourself, we'll go on a lovely ride. So I'm going to give some refresher, or maybe it's new information on what sequences are for you. Briefly touch on the methods of sequencing and the different sequence file types you might run across. The analysis methods that are used for sequences that'll come into play with when we talk about demonstrating an analysis program for you. We're going to have a part where if you want to at home, you can make your own FASTA. I've provided a shared folder. There should be a link to a Google Drive folder that you can download those FASTA files, and then the document that you can use to make your own. And then I'll demonstrate how to do the analysis in Mega, which is a free program. You do need to go yourself and download it. It's not something that I can distribute. And there should be a link to the Mega software website. And then I'm going to demonstrate, depending on how much time I have left, some of the web-based sequence and analysis resources that you can access, both freely available and then University of Minnesota VDL tools. So first of all, what's a sequence? It's pretty straightforward. It's things placed in order. Usually when we're talking about pathogens, we're talking about typically a nucleic acid sequence, and that could be either DNA or RNA, depending on the agent or what you're looking for. Most RNA agents get translated into DNA for sequencing, and you end up with the DNA code. And so that sequence will look like a string of characters, usually corresponding to the nucleic acid bases, so A, C, G, T, and U. Occasionally you may run across some others, so there are several other characters that you may see. So an R would indicate that it's hard to tell, we can't tell them, but there might be an A or a G at a position, so that's a mixed base. And so it's not necessarily strictly just A, C, T, and G or U. And if you run across it in a sequence file, you see a dash. That means that there's a gap. So when that was assembled, probably to a reference genome, there was a gap present. The other sequence that commonly gets analyzed are amino acid sequences. Those are translated from the nucleic acid sequence. And those are also single letter codes, but the A's and the C's are going to be different. So it's important to know what you're analyzing. So A would be alanine rather than adenosine. And so the type of sequence you're dealing with is important to know. So how do you get them? One of the most common methods is Sanger sequencing. That's one of the early methods. It's called chain termination. So it's PCR based and uses a known target. So you have to have designed primers in order to find what you're looking for. But it does have the advantage of being PCR based. You can amplify very small amounts and get to known quantities of whatever amplicon you're looking for. The other part, which Sunil touched on, was the next generation sequencing. This is NGS is a catch-all term. There are many different technology platforms that are used for next generation sequencing. Typically what it is it's the next generation after Sanger, and it means that it's using a shotgun or a metagenomic type approach. And when I talk about technology platforms, it might be short reads. When I say short reads, I mean it could be 75 base pair reads that it's looking at, maybe up to 500, somewhere around there. Or it could be long read technology where you're looking for thousands of base pairs at a time. Those might be Illumina, it might be PacBio, it might be MinION. So there's a variety of things that get lumped into next generation sequencing, and they're not all the same, and they don't all have the same characteristics. We can, as Sunil mentioned, combine this next generation sequencing technique with PCR preamplification, and then you end up with a much more robust targeted sequencing approach if you know what you're looking for. So coming off of the sequencers, there's different types of files that you may run across or see. So Sanger sequencing, you typically will get an AB1 file, and this is called a chromatogram or a trace file. And I'll show you examples of these in a little bit. The next generation sequencing or NGS files are more complicated. They will have quality associated with them. They'll have lanes associated with them. So there's more information. And so those typically will come off as FASTQ files. They may be, because they're very large, they may be zipped. And so you may see a GZ at the end of your FASTQ. So here's an example of an AB1 file or a chromatogram. This is showing what the sequencer would identify at these different positions. So the green peaks that you see here correspond to the T reporter in the PCR reaction. This yellow line is the G reporter here. We have a red reporter, this would be A, and the blue reporter is C. And so as this chain terminates at the different lengths, the machine will read what nucleotide got added, and it'll create this profile that's called a chromatogram. And so this is where the A's and the C's and the T's and the G's come from when you look at the typical file that you're used to looking at. For NGS or the FASTQ files, there's a little bit more information there. These are called kind of the raw reads. This is a subset of a FASTQ file viewed in a genomics program. And so this initial part here is the identifier. There's a lot of information here that doesn't mean anything to me. It might be telling me what type of machine it was done on, what lane it was in. Then there's the sequence itself. And each one of these bases will have a quality call. And so that would be all part of the FASTQ file that would be spit out of the machine. And you might potentially see if somebody sends you a sequence file. So once you have those traces or those sequence files, how do you get to the full 603 base pairs for PERS or 5, or the 16,000 base pairs for a PERS whole genome? And so this comes down to assembly. And so this is an example, a depiction of what a reference base, so there's a reference right here, assembly using Sanger sequences. So each one of these, they're AB1s. These are the trace files, and they're stacked up on top of each other and compared along each position to see what base is in there. And you notice that many of these are not the full 603. They get chained together to create the full sequence. And so this would be an example of how a Sanger sequence would get assembled. Ideally, you want 2, preferably more. So in this case, we've got 4 coverage of 4 right here in that 500 base pair, or the position 500 to 600-ish. But then we drop down to 3 for the rest of the sequence. And this would be good. If you're down to single coverage, you're not certain if anywhere you see these blue marks would indicate you have poor quality. So you don't know for sure if what a single call is, whether or not it's true. So this is what an example of a de novo assembly using NGS data would look like. And so each one of these bars, rectangles that are stacked up here correspond to reads. These are 150 base pair reads, and the computer algorithm will take and try to line up all of the A's and C's and T's and G's together and stitch together which ones might be next to each other using proprietary algorithms or open source algorithms to do that. You'll notice that the depth is much different. So here on the left is the depth chart. So here in this 600 to 700 base pair range, we've got almost 25 reads covering those bases. And that's much, much better than our 4 Sanger sequencing. And we also have some regions of this contig here that have low coverage as well. So you may be a little bit cautious to say if this base calls here where you have 2 are real or not. So I showed you kind of the different ways to make the sequence so that it's been assembled. What typically will come back to you? Usually what sequence files are coming back would be FASTA files, .fasta or .fas. Sometimes they're shortened. And so these are very, very highly structured files, and they have a specific format. There's 2 parts to it. So there's going to be a description line or a def line, and that will always start with the greater than sign. So the little arrowhead that's pointing to the right there, that line will contain any information that you want to identify the sequence. I always advocate using an informative naming convention. So if you're going to make your own, you would put what identifies that sequence to you. So here at the diagnostic lab, it would be our case number and sample number. Maybe we'd put what sample type it came out of. I'd want to specify, so if I'm doing PERS, I would say it's PERS. Albert talked about the fact that you can have a whole genome, or you might be having OR5, so I would put in there that this is PERS OR5, and that would tell the program that I'm going to use what sequence I'm dealing with. It'll also help you, if you open that file in a text editor, to know what you're looking at. And then the second part of the FASTA file is the sequence lines, and that can be nucleic acid code or amino acid code. Those sequence lines most technologies can handle full length, so they can run for thousands and thousands of characters and they don't have a problem. Early machines had issues with how much they could display, so convention sometimes says that you stop a FASTA at 80 characters or 120 characters per line. That's not necessary, but as long as you don't put this character in the middle, you can hit enter as many times as you want to create new lines so that you can read what's in your file. FASTAs are typically just one, or you can lump related sequences together. So a multi-FASTA file would be labeled just the same .FASTA, but in that file there would be several sequences, and so that would be an example of maybe you've got 5 PERS or 5 sequences from one farm that you want to maintain and use in comparisons later. You can create a file that has all of those together. So here's an example FASTA file. At the top we have the def line. There's an arrow. It tells you what it is. It's PERS. In this case I just made it up or anonymized the sequence, and so this is just called new. This would be an example of if you got a new one at your farm and you want to know where it fits, you need to identify that it's new. You don't want to just leave it as PERS, because all of your other PERS will be labeled as PERS something. So the FASTQ, this is somewhat similar to a FASTA, but it's a little bit different. It provides quality information. The def line doesn't start with a greater than sign. It starts with an @ sign. Then you do have sequence, which would be typically your nucleic acid codes. There's a plus sign. That can have some of the definition information after that if you want, and then there'll be a quality line, and those quality codes would correspond with the particular nucleic acid that would be in the same. So these sequence line and quality line need to be of the same length so that they match up. Here's an example of what a FASTQ file would look like if you open it up in a text editor. So this @ sign shows the def line, so this is defining what this is. We have the sequence that follows thereafter. There's the plus sign to let the program know something else is coming. And then these characters right here are ASCII characters that are telling the software or the program what the quality of these bases are. So this arrow or this greater than sign is not actually starting a new line. That's an ASCII code that corresponds to a specific quality call. This looks like a foreign language to me, but the computer knows what to do with that information. Another one that you'll see if you're using Mega, some softwares will allow you to export it in Mega. It's just another way to format it. It's similar to FASTA, but has more information that you can provide. This will always start with a hashtag and then Mega. And then The additional lines have that are giving you additional information will start with an exclamation mark, and you have to terminate those with a semicolon. You can put in their title, you can put in description, and sometimes it'll have format, so it'll tell the program that it's a DNA sequence. Then there will be sequence lines that will be started with a hashtag, whatever your identifier is. So, hashtag PERS-new, and then the new line that follows thereafter would be your sequence line. You can also include several sequences in the same file. After this quality information, you would have as many hashtags as you need to create those multiple sequence files. So this is an example of what a mega file would look like. If you open it up in a text editor, you have hashtag mega, you have exclamation point title, so telling the program what the title is going to be. The title is PERS-A in this case, and then we have the defining part for the program. So when it takes the sequence and uses it, it's going to call it PERS-A, and then we have the actual sequence itself. So once you have those files, the big question is what to do with them. We get the requests quite commonly. Can you analyze this sequence for us, or can you sequence that so I can analyze it? What is all involved with that? The primary analysis revolves around alignment, and alignment is, strictly speaking, just arranging the sequences relative to each other. A pairwise alignment is fairly easy to do. You could do that by hand, but when you start combining multiple sequences together, it can become difficult very quickly. And so this would be an example of 8 influenza A virus PA segments that have been sequenced. These are unaligned. When I look at them, I notice that at the tail end, they're slightly different lengths. Well, when you look at that, we're not sure if the start of the sequence that these would map to starts at number 1, or would it start somewhere else. And so when we align them, we start to find out, well, this sequence number 1 was actually a little bit short in the beginning, and so it shifts to the right as far as relative to sequence number 2. And so this would allow the program to figure out where position number 5 is, or 150 for all of these sequences might be. And this is an important step for the analysis. So once you have it aligned, then it becomes a little bit more important to try to tell how are they different. So we know that these sequences are different lengths, but this graphic isn't showing you any additional information. So this image right here is showing you differences. So this is the same alignment, but now I've added in the ability to see where there might be polymorphisms or changes in the sequence. And those are corresponding to the black lines. If you see gray, it means that it's agreeing with the consensus, which would be, when we look at these sequences here, which ones hold the same value, or what's the most commonly found base in that position. So the consensus is important, and then the change from consensus is shown here with the black bars. It doesn't really tell you what the change is, and so in order to do that, you have to go up a little higher and look at higher resolution. And so these are the same 8 samples, but looking at the bases where we saw some of those black tick marks, and here we start to see where the differences between these sequences lie. So when you see a color change, that means that the sequence is different from those around it or those below it. And so there are some that seem to be carried through. So these first 4 sequences, you'll notice that in position 540, they all have A's, and the second 4 sequences in position 540, they all have G's. And so there are times when you see large groups that have the same mutation, but then when we look in this, the first 4 besides that are fairly similar to each other. When you look at the second group of 4, 5 through 8 down here, you'll notice that there's a smattering of changes that are not necessarily conserved. A lot of them have this C here back at the maybe the 515 base, but then there's these T's and A's that are kind of scattered throughout. And so the question then is trying to figure out, did one come from the other, or what's the change? How did the sequences change to arrive at this point? When we talk about the changes, we would call those mutations. I'm talking about just the kind of the basic categories. The ones that I depicted here in this image are substitutions. So you've got a G that changed to an A, or an A that changed to a G. Substitutions have a variety of. There's a few different ways that substitutions can affect or not affect the organism that the sequence is within. And so in some cases, when we think back, I made a big leap and I didn't explain it. We would say that the nucleic acid is going to get translated eventually into amino acids or proteins. And so the amino acid takes a 3-block chunk, or a codon, of genetic information, DNA, or in this case it's RNA, would then correspond to a specific amino acid. So sometimes there might be multiple codons that give you the same amino acid. So if this change right here, a G to an A, doesn't change the amino acid on the end product, that would be called a silent mutation. If you've got one where this A to G changes the codon reading and it changes the amino acid, that would be called a missense mutation. And then if for some reason this change, maybe the C got changed to a T, you can make a nonsense mutation. So that would be a stop codon, and that would terminate your protein before it was supposed to. Other mutations would include insertions or deletions. And so those might be just adding one base. If you do that, you get a frame shift. You start changing the way that the codons are read. So the positions prior to that would have similar amino acid composition, but after that you wouldn't necessarily see it. It would start to change that. If you add in or delete a 3-block chunk, you don't get that frame shift. You would just have either an extra amino acid in the amino acid sequence, or one less. But the chunk prior and the chunk after are still going to be homologous to the wild type. So how do you analyze these differences? I talked about the fact that you can look at 2 sequences by yourself and you can align them. That's not a huge problem. But when you start adding 10, 20, 50 sequences, it gets a bit difficult. And so there are alignment algorithms. There's a variety of them. Some of the names that you might run across would be Clustal. The latest version is Clustal Omega, but Clustal W is still widely used, so you'll probably see that if you're doing any work there. Muscle is another algorithm that's used, and then there are proprietary algorithms within specific softwares, typically those for purchase. So Genius might have one, CLC Workbench might have one, LaserGene might have one, where they're probably using some of the same concepts, but they're applying it a little bit different in their software. And these algorithms can use different methods. So progressive you'd start with a core and then get those aligned and add to it. Iterative would be you take a bunch, you stick them together, see where they fall, you do it again, and then slowly over time, as you do the process, would get those alignments to match up with what reality probably is. So after it's aligned, what's the output look like? So Albert showed the heat map. This is a similar. This is those same 8 PA segments of the influenza virus that I was using in the other examples. And so in this case, the darker color black is closer together. You might see this right here is an identity matrix. So it's showing the % identity between the different samples. You take the row and the column and you match it up. So sample 2 and sample 1 are 99.7% similar. A distance matrix would just be 1 minus this value. So in a distance matrix, this would be 0.03-ish. So 28, I guess, based on the significant figures there. So just know what program you're using, what it outputs. So this tells you pairwise what the differences are, but it can be somewhat difficult to know if they have very similar % differences. So you'd think, oh, number 1 and number 2 are both 99.4% similar to sample number 3, so they must be closely related. you don't have a guarantee that these two, even though they have a similar percent difference or percent similarity, that they themselves are necessarily close to each other there. So it makes it a little bit more difficult. So another way to look at it would be at a phylogenetic analysis. And so this is trying to look back for a common ancestor. So the idea would be looking back to see where there might have been a shared viral strain or bacterial strain or whatever it is you're looking at that gave rise to these different populations. So when we look at those same 8 PA segments for the influenza virus, we can see some clusters. So there are these right here are when we look at the distance you look at on a phylogenetic tree is not necessarily how different it is from top to bottom, but it's from left to right. So here, these are on the same line virtually, so they're virtually identical. They're not that far off from these other tips right here, so they're still pretty close to sample 2 and sample 3, but there are some differences. Then if we add that up, so down here at the bottom, there should be a scale bar on all of your phylogenetic analyses you look at. That scale bar will tell you the percent differences there. And so you would add those factors up. So the distance from this here back over here would tell you, oh, this sample right here is these 6 and 7, this little cluster here is much different than these 1 through 4 that are up here. And then you've got number 5 and number 8 that are yet even more different. They don't share a common grouping in this particular sequence set. It's important to know too that most of what I discuss is phylogenetic analysis. So these are looking for common ancestors. Occasionally you can get dendrograms. That means that there's not necessarily an evolutionary component to it. It just means it's looking at percent similarities. Those would not be looking at necessarily or give you an idea of how related evolutionarily the organisms are that you're looking at. So this is a phylogenetic tree. Sometimes you'll see cladograms. Sometimes they'll change the meaning of the bars here. I'm going to keep it simple and talk about the phylogenetic analysis where you have a tree that would correspond to more of like an evolutionarily type analysis. So when we look back here at this, we have the choice in this case. I did nucleic acid analysis. We could have done amino acid analysis. So the question comes sometimes is, why would you use a particular sequence type? And so nucleic acid is most useful for epidemiological investigations. It has the highest resolution to be able to find changes. So we talked about the fact that 3 nucleotides correspond to 1 codon or 1 amino acid. And so if you have a silent mutation, the amino acid sequence will not pick that up, so you'll lose that resolution of change. But on a nucleic acid sequence, that change, even though it's silent, it doesn't necessarily make a large difference for the organism, would still be a point for you to try to differentiate 2 different organisms or strains. From the amino acid sequences, that would be translated from the nucleic sequence. It's pretty straightforward in most everything that we look at. Our bacteria and viruses are pretty simple, and so they don't have introns and exons. If they've got a coding sequence, the whole thing is gonna go. In humans or complex organisms, it gets a little bit more challenging because you have segments that are coding and some that aren't coding that are part of the same gene. We're not going to get into that, but just keep that in mind that we're dealing with very simple things. If you talk about genetics, it can get complex real fast. So amino acid sequences are good for functional analyses. So if you want to know if the protein is going to remain similar or maybe have the same activity, that's where an amino acid sequence would come in. Immunologic assays, the immune system just cares about what protein is being expressed on the outside of the cell, and so it doesn't care if there's a silent mutation. It just wants to know whether or not the protein is different. So if you're looking at trying to figure out is this similar or not similar to a vaccine strain, that might be, so for example, influenza virus HA sequence. That one might be one where you'd want to do an amino acid sequence to look at if you're trying to figure out if a vaccine might work or not work for you. You might use the nucleic acid sequence for on your farm to try to figure out are things changing over time. So now we've come to the, as Jerry said, it's my part without a safety net. This is the hands-on or demonstration part of it. So I don't know who has or hasn't done this type of thing. Oftentimes I get a query that, can you analyze the sequence? And I say, sure, send me the FASTA file. And I get a diagnostic report or something similar. So I'm going to walk through how to create your own FASTA file. It's fairly simple. The pieces that you're going to need are the anonymized diagnostic report that I have in the Google Share folder. And then to create your FASTA file, you don't need Mega yet, but if you haven't downloaded that, that should be showing up in the chat, the link to Mega. That'll be the next step. And so the software you're going to need is a text editor. You can use Notepad. That's built into Windows. If you're using a Mac, I guess my advice to you is to buy a Windows machine. Hopefully Apple's not listening. But I'm not sure. There should be a text editor similar to Notepad built into your iOS. There are other programs out there. I like Notepad++. It gives me line numbers and helps me type things out. But for this demonstration, we'll do Notepad. So without further ado, I'm going to pull up. We'll pull up the example anonymized report. And so this is an example. I've ripped out the case number. I've ripped out the owner. What we have is a serum case that had a positive PCR result. When we look at this is our summary report. We run the test for 40 cycles. The sample had a CT of 30. So this is up at the upper limit of what I would recommend trying to do Sanger sequencing on. Normally I try to use clean samples. So serum, if you're talking about flu, lung, or nasal swab, oral fluids tends to be a dirty sample. There's a lot of enzymes in there that'll break down your nucleic acid. And so be cautious of that. This is up at the upper limit of what you can expect to get a sequence back out of, but a PERS sequence was obtained from that. So here we've got a PERS sequence. We give you some information telling you about what we sequence PERS R5. We remind you that this RFLP that folks like to look at doesn't tell you how related something is, and to look at your sequence to actually tell if they're related. For VDL clients, we do maintain, if people would like us to, we do maintain client charts, and that's helpful for large sequence bundles, or if you don't feel comfortable doing it yourself. So sequences that are done here, you just need to contact us, and we can set up charts for you based on your sequences you've had done here at the diagnostic lab. We can also do analysis on other sequences from elsewhere. There is a small fee associated with that, so we can maintain your client chart with external stuff. I'm hoping here you'll feel comfortable being able to do some analysis yourself. So here we have a sequence. It's all by itself. You say, Matt, how do I get this in FASTA format? So we recall back, there's a specific way that you structure these. So the first part is you need to get your text editor open. So here I've opened Notepad. If you have a Windows machine, you might need to go to Windows Accessories to find Notepad, or just type it in your search bar. And here I'm going to start creating the FASTA file. So the FASTA file needs to start with the caret, or the greater than sign, and then it has to have some identifying information. You can put whatever you want in here. I'm going to call this PERS FASTA I made myself, if I can type. All right, so this would be my def line. This is going to tell the program what the sequence is. You hit enter so that you're down to a new line, and then you go to your document that has the sequence file or the string of characters. So if you're in a PDF, you can select it. you copy it, so Control C, or you can right click and copy, and then you're going to go to, now that I remove my taskbar for this, it's hard to find stuff, and then you would paste that. So I'm gonna push Control V, or you could just do paste here. And so what that does is it puts the sequence in this file. It's still not saved, but it's in the right format. It doesn't matter that they stop after a certain period. There's no blank lines in here, and I didn't put a caret anywhere. But just be careful that something extra didn't get carried in that you would need to potentially delete. So at this point, we're gonna save the actual file. So you go to File, and then you click Save, and it should prompt you to do what type of file do you want to save. So pick a spot. I put downloads here. And then the other important part is that if you leave this as the base, which is .txt or a text document, it's not going to work. And so I think I'm going to have to switch my microphone here just a second. I can hear something beeping in my ear. All right. Hopefully that's going to work for you. I hope the quality is going to be adequate. Can you still hear me? Okay, Ralph says it's okay. All right, so I do apologize for any changes in quality. The yeah, no, it's not as good. It's off my webcam. So the point here is that you can't leave it as a text file. So in this Save As type, you would drop down and select All Files. And then you can specify the file type when you write your file name. So in this case, I'm going to call it PERS I. So I get in the convention of I don't put spaces, or I try not to. It depends on what programs you're using, whether or not they handle them. So we'll just PERS I made. And then the important part here is that you put .FASTA. So when you do that, that will tell the system, because you said on here save as all file types. You're saying I'm going to save it as a FASTA file, and that'll specify it. You need to be careful with like Word documents, using a rich text file editor. They may corrupt it. So that's why I use Notepad or some other basic text editing software. So you click Save, and once you've done that, you can still see it in the Word document, but now you've created a sequence file that can be utilized by an actual sequencing program. And so that's the part of the show where we talk about creating your own FASTA. Hopefully everybody was able to follow through on that. The next part would be to create an alignment. So there are example documents in that shared folder of several PERS. I don't know that I'm going to have time to go through all of the things I was planning, but we should be able to get through some of those. And then the software you're going to be using is Mega. So again, look in the chat for those, and go from there. We'll bring up Mega here. So this is when you open Mega up, this is what it's going to look like. It's fairly blank. There's the way to get data in here is you can either click Data on the button here, or you can go to File, Open a File /Session. And so if you click on Data, you can get the same options here. You can click Open a File or Session. So you're going to go to the Downloads folder, or wherever you put your file, the files there, and you can open up the different FASTA files that I provided for you here. This PERS I made, just to prove that it's actually a FASTA file. We can click Open, and it'll give you the option. Do you want to align it with something, or do you want to analyze? In this case, I'm just going to click Analyze to show you what the file looks like. I'm going to tell it it's a nucleotide sequence. I'm going to click OK, and then it asks you if it's a protein coding nucleotide sequence. If you click yes, it just asks you to tell you what kind of organism it is. So you would say it's just the base translation or there's some specific. So I just typically click no. And then you're like, well, I opened it. Where did it go? The important part here in order to see what you're looking, what you opened is to click on this TA button here. This is going to open your data file. It might be in a different screen like mine was. And so I'm going to change my font here so that hopefully it's readable. And so here, what you see is the sequence. And so it puts that information that you put it after the caret. And then this is the sequence above. We can change this so that way we don't see those identical symbol. And then it'll put the sequence down below. So if you have multiple in here, it'll put a consensus at the top. And it'll put a dot where it's the same as the consensus, and it'll put the character where it's different from the consensus. So that's just the reason why that shows up like that. And so you've now made your own FASTA. It's readable by Mega, and that's a first big step. So in the case of you want to make an alignment, let's see here. No, I don't like to save stuff. Just start from scratch each time. You'll figure out what works for you. So in this case, I want to make an alignment. So I'm going to click the Align button. You can do Edit or Build, or you can do Open a Saved Alignment Session if you already have one open. So in this case, I'm going to click Edit or Build. You have the option of creating a new alignment. That one isn't as good. I would click the Retrieve a Sequence from a File. So if I click OK here, there is this list that says PERS list. So if you click on that and click Open, it will open in the data viewer or the Alignment Explorer right here. And so this is an example of a list of, I've got 4 different PERS sequences here, PERS A, B, and C. And so the colors show you, kind of give you a graphical idea where something might be the same or different. When you look at this, they all start at 1, and we look down to the right, to the far end. Hopefully you're not getting too dizzy. Oh, wait a second, there's some here. There's one that's longer and a couple that are shorter. So this would indicate that maybe they're not actually aligned yet. So this would become where we're going to do the alignment and figure out where things are supposed to line up. So you can click on alignment and say align by cluster W or align by muscle, or you can click on the W, which is cluster W, or the muscle picture here. I typically will do muscle because it's faster, but you can do what you like. And you would click what you're going to do, align DNA or align by codons. So I'm going to click align by DNA. I forgot to select the options over here. So in order to select those, you need to push Shift and click on them. Otherwise, if you want to do all, you can start it and say select all. Click OK. And then it's going to open an options button. There's gap penalties, how many iterations you want to go, and some of the other advanced options. That's beyond the scope of today's conversation. Just click OK. And then what you end up with is a file that shows you where you might have some differences. I think maybe I actually had aligned this. But anyways, the idea here is you're going to get all of the ones that can line up to line up. And so hopefully at position 603, that would be position 603 for all of the sequences. And so this would be how you create an alignment using Mega. You can add sequences to this. So you've got A, B, and C, but oh, you got a new one. The question is, where does that new one fall? You can click Insert Sequences from File here and add another one. So we're going to add PERS new in here and click Open, and it'll slot that new one in here. You'll notice that it doesn't have the same, it may not necessarily be aligned yet. So once you do that, you would click your Align again, click Align DNA, select all, click OK, and it'll line it up for you there. So that's the alignment. And I'm going to check my, let's see here. Yeah, so one last thing in Mega before we leave Mega behind is you can also create trees. And so that would be, let's close this down. I don't want to save that to the file. This phylogeny button right here, it looks like a tree. You click on that, and you say you want to construct a tree. You can do a maximum likelihood. You can do neighbor joining. You can do minimum evolution. You can do neighbor joining tree. They're quick, and it asks you for the data file. So I'm going to look for data files. It asks you to look for it. I need to find my FASTA file. So we will do the PERS list here. Asks you what is it? Is it a protein sequence or a nucleotide sequence? This is a nucleotide sequence. Click OK. No, it didn't like. Where did it find an extra character? We'll create a new one. So you should be able to do an alignment, build, retrieve it from a file. In this case, we're going to open several of these at once and create a our alignment again. So now when I have the alignment open, I come to the phylogeny and I say I want to create a neighbor joining tree. I'm going to select all those, forgot to do that. We'll align the DNA. We'll do our fun stuff. All right, I'm going to minimize this, and now it should allow me to, we'll just save this. This is the safety net that Gary was talking about. All right, now when I do this, should be able to look up an alignment. And so in this case, it's going to bring up options. I just click OK. You can go through and change the models. It's not really necessary. It'll run the analysis for you, and it'll create a tree for you. So this is an example of a tree. So PERS A and B are virtually identical. They're on the same bar, and then PERS C is a little bit different. You can add information or see below. There's a view. Should be an option for the caption to tell you what the branch length means. And so that would be how you would create a tree. You need an alignment, and then choose which type of tree, and you can spit it out and figure out which one is graphically closer to each other. The other thing that you can do is you can create the distance matrix. You can do the pairwise. So yes, I want to use my active data. I'm going to do an analysis, and so this is a distance matrix. So they're very close to each other. 0 means that they're identical. The 0.25 would indicate that they're slightly different. And I do apologize. I see a comment in the chat that couldn't get to the Google Drive. You can contact me directly. I'll provide my email after this, and I can provide those files for you. All right, so one other part would be identifying an unknown sequence. So the documents you're going to need here would be the example unknown FASTA file, which would be in that Google Drive folder that apparently people can't access. And then you're going to need to go to a website. So the website is NCBI BLAST. There's also a BLAST option that's built into the alignment viewer of Mega. So I'm not going to go through that, but you can, if you have the alignment viewer open, you can blast certain things out of the alignment viewer. So we'll open up BLAST here. So this is the BLAST website. NCBI is the National Center for Biotechnology Information, and BLAST stands for Basic Local Alignment Search Tool. This is the way to look through databases. If you need to figure out what a file is that you're not sure what it came from, you would use nucleotide BLAST. So you click on here, and then it's going to give you an option, right? So you can either paste in your FASTA file, or you can upload a file. In this case, I'm going to upload a file. Most people will have saved it, or you can paste it in and just make sure you remember that you have the caret there. So in this case, I'm going to click Choose File, and I'm going to go to my downloads again. So in this case, there should be an unknown sequence that's part of the set. It's a FASTA file, so it should be good to go. I'm going to click Open. It pops up here to say that it's there. You can go through and change which database you're going to look against. And so the options, you can play around with these, but the important thing is you have the file either pasted in here or uploaded, and then click BLAST. And so what NCBI does is they look against the entire database of all the known nucleotides that are out there, and they try to find things that are similar. And so in this case, I get a report back that says sequences producing significant alignment. So it usually gives you 20. And when you look at the names, this tells you that these are all Streptococcus suis. So now, instead of having a sequence file that I don't know what it is, I don't know what it corresponded to, I now know by using BLAST that it's a part of a Strep suis genome. Some of them will tell you maybe what particular gene it's associated with, but in this case, it's just sending you to a Strep suis genome. So that would be how you would use an online example of how to identify the unknown. The classifying unknown, I'm not going to waste time here. There's the influenza research database. It wasn't working this morning when I tried it. It can be temperamental. I put in here. They have a good database of influenza specific stuff that also may be curated. You can put your own in a workbench there. I am going to show you the ISU, Iowa State University CVM's Flucher website. So this is a good resource for people that want to know a little bit more about the flu sequences that they get back. It's specific to H gene, as far as I know. Maybe there's N gene in there now, but the example influenza A virus FASTA file is in that Google Drive folder. So I'm going to pull up the influenza. So this is the influenza research database. If you're going to use it, click the Analyze up here, and then do the Identify Similar Sequences, the BLAST. And then you would go through, you can either click the option to paste it in, or you can upload things. And then you need to specify what database you're going to use. So influenza genes or whatever sequence segment it's from. So let's pull up here. So the ISU Flucher website has a HA identity tool. If you click that, you can paste in your sequence. So I'm gonna open up the example flu that I had. So here I've got my notepad. I'm going to select it all. I'm going to copy it. I'm going to paste it in here, and I'm going to click search. And so what this will do, it'll tell me the clade match. So this is an H3 virus, 2010.1. So this is a human-like virus that is endemic in pigs. And so also included are things of when it was last isolated or something similar was last isolated. So ISU VDL has one back in November of last year that was similar to this. And that, I was hoping to get to our Lab Savvy program, but I am willing to follow up with anybody that has a desire to follow up with Lab Savvy. This is specific to your VDL client, so you do need to have login privileges there. So the take-home message here, I hope you understand the foundational aspects of genetic sequencing and analysis. You now can create and modify your own FASTA sequence files, and you can use the basic functions of a free to download desktop genetic software, and hopefully now you have some basic understanding of some of the online genetic sequence analysis tools. I do need to thank especially the molecular diagnostic staff. They produce all of our sequences. And if you got any questions, you can put them in the chat or email me directly.
Thanks, Matt. We put you up on the high wire, and then we threw stuff at you by shortening your time. So that's the exciting part of a live presentation. You did a great job. I think we'll go ahead and take our 15-minute break at this point, and so we look forward to having all of you join us again at 10:15. And thank you for all the presentations before. The next session will be talking more about bacterial genotyping, NGS work, in situ hybridization, other applications besides viral genotyping. So I think it will be interesting, and we look forward to seeing you again shortly. So thank you.